I am going to discuss on the topic cellular organization of the pituitary hormones and their function. The pituitary gland or hypophysis is often called the master endocrine gland because it secretes many hormones that can influence the action of numerous peripheral tissues or organs in the body. However, the pituitary gland itself controlled by the hypothalamus of the brain, from which regulatory hormones are transported to the pituitary. The hypophysis is a protrusion of the bottom of the hypothalamus at the base of the brain and rests in a small bony cavity, the cavity known as sella turcica, that is covered by dural fold. The pituitary is functionally connected to the hypothalamus by the median eminence via a small tube that is called the infundibular stem or the pituitary stalk. The hypophysis has two functional components. One is anterior pituitary, that is known as adenohypophysis, that is a glandular epithelial tissue, and another posterior pituitary, that is a neurohypophysis, that is a neural secretory tissue. Now, what is the embryologic development of the hypophysis? The structure and function of the hypophysis reflects its dural embryological origin. During embryonic development, the epithelium of the pharyngeal roof forms an outpocketing that is known as hypophyseal pouch, that is a Rathke pouch. As development proceeds, the hypophyseal pouch detaches from the oral cavity to become the cellular or glandular portion of the hypophysis that is known as adenohypophysis. At the same time, the down growth from the developing brain forms a neural portion of the hypophysis, that is a neurohypophysis. These two separate development structures unite to form a single pituitary gland that is known as adenohypophysis. The hypophysis remain attached to a ventral extension of the brain that is called the hypothalamus. A short neural stalk, that is the infundibulum, that is a neural pathway that attaches and connects the hypophysis to the hypothalamus. The neurons that are located in the hypothalamus that control the release of hormones from the adenohypophysis, as well as secrete hormone that is transported and stored in the neurohypophysis until needed. Now, the hypophysis is categorized into two portions: anterior pituitary, that is adenohypophysis, and the posterior pituitary, that is a neurohypophysis. So that is the main divisions. Now the adenohypophysis is further divided into pars distalis, pars tuberalis, and pars intermedia, whereas the pars distalis and pars tuberalis, that forms the anterior lobe of the hypophysis. The neurohypophysis is divided into pars nervosa, infundibulum, and median eminence, the pars intermedia and the pars nervosa that form the posterior lobe of the pituitary. Now, so first one, the pars distalis, that is a larger part. That is a larger part, and that contain two main cell types: chromophobe cell and the chromophil cells. Then pars tuberalis, that portion is the pars tuberalis that surround the neural stalk region, and then pars intermedia, that is pars intermedia, that is situated between the pars distalis and pars nervosa, and that contains a follicle. And you see here colloid field cystic follicles that's aligned with basophil cells, and it is remnant of the humans. It is a very rudimentary structure. Then you see here there's a pars nervosa that is characterized by unmyelinated axons and the supportive tissue, that is a pituicytes that contain oval shaped nuclei. And infundibulum, it is a neural stalk that connects neurohypophysis to hypothalamus. And the last one, that is a median eminence that is located at the base of the brain. A connective tissue capsule that surrounds the pars distalis and pars nervosa portion of the gland. Both the pars distalis and pars nervosa are supplied by numerous blood vessels and capillaries of different sizes. Now, What are the cell types present in the adenohypophysis? The cells of the adenohypophysis are classified as chromophobes and chromophils based on the affinity of their cytoplasmic granules for specific stains. The chromophobes that are so named because they are not stained with dye. These cells are pale stained, have less cytoplasm than chromophils, and do not possess any secretory granules. And then chromophil cells that are so named because they have affinity to dyes. It may be acidophil cells, it may be basophil cells. The acidophil cells, that is also known as alpha cells, and the basophil cells, that is known as a beta cells. The acidophil cells, again, characterized into two categories: somatotroph cells. The somatotroph cells basically stained with orangey and agent stains, whereas the mammotroph cells that are stained with azocarmine cell. So acidophil cells are classified into two types, somatotrophs and mammotrophs, whereas the basophils are categorized into thyrotrophs, gonadotrophs, and corticotrophs. The thyrotrophs that are stained with aldehyde fuchsin stains, whereas the gonadotrophs are PAS positive and corticotrophs are also PAS positive. Now, the hypothalamic releasing and inhibiting hormone that are secreted near capillaries at the base of the hypothalamus. The capillaries drain into short blood vessels that are called the portal vessels, which subdivide into secondary capillary bed within the anterior pituitary. On reaching the adenohypophysis, the hormones binds to specific receptors on the cells, and then they stimulate the cells to secrete and release of the specific hormones into the circulations or inhibit the hormones. Then you see here, there's a hormone that is secreted from the anterior pituitary: FSH, TSH, ACTH, prolactin, MSH, and GH. Anterior pituitary is an endocrine gland that synthesize and secrete hormones in response to signals from the hypothalamus. The hormones released from the chromophil cells that are carried into bloodstream to target organ, where they bind to specific receptors that influence the structure and function of target cells. Once the target cells are activated and release their secretory products, a feedback mechanism, it may be positive or negative, that controls the synthesis and release of these hormones. That is interesting. Now, the hormones of the adenohypophysis, they are categorized into two main groups according to the target tissue. Some group exert their principal effects on the non-endocrine tissue, as for example, growth hormone that influence growth and nutrient metabolism in tissue such as bone and muscles, and other hormones are prolactin, melanocyte stimulating hormone, etc. The second group that includes hormone that control other endocrine glands. By convention, hormones that influence the function of other endocrine glands have the suffix tropic in their names. They are called tropin hormone, thyroid stimulating hormone, adenocorticotropic hormone, luteinizing hormone, follicle stimulating hormone, etc. Now, what are the cells and hormones of the pituitary? The first one, the somatotrophs, they are also known as orangiophils or alpha acidophils. They secrete somatotropin or growth hormone. This hormone targets the whole body and its general growth. It stimulates cellular metabolism, uptake of amino acids, and protein synthesis. Somatotropin stimulates liver to produce somatomedin, that is also known as insulin-like growth factor. Somatomedin influences cartilage cells in epiphyseal cell plates to increase growth in length. Growth hormone inhibiting hormone, GHIH, that is somatostatin, inhibits release of growth hormone from somatotrophs. The next category, mammotrophs. They are also known as lactotrophs or carminophils or acidophils. They produce lactogenic hormone, prolactin, that stimulates the development of mammary gland during pregnancy. After parturition, prolactin maintains milk production in the developed mammary gland during lactation. The release of prolactin from mammotrophs is inhibited by prolactin release inhibiting hormone, that is dopamine. Then thyrotrophs secrete thyroid stimulating hormone thyrotropin or TSH that stimulates thyroid gland hormones. TSH stimulates follicular cells in the thyroid gland to synthesize and secrete thyroglobulin and the hormones thyroxine and triiodothyronine from the thyroid gland. Next category, gonadotropes. They are also known as signet ring cells. They secrete follicle stimulating hormone and luteinizing hormone. In females, follicle stimulating hormone promotes growth and maturations of ovarian follicle and the subsequent estrogen secretion by developing follicle. In males, follicle stimulating hormone promotes spermatogenesis in the testes and secretion of androgen binding protein, that is ABP, into seminiferous tubules by Sertoli cells. The androgen binding protein maintains the needed concentration of the testosterone on the seminiferous tubules to ensure proper spermatogenesis. Now, in females, luteinizing hormone induces follicular maturation, ovulation, and corpus luteum formation. Luteinizing hormone promotes the secretion of estrogen and progesterone from the corpus luteum. In males, luteinizing hormone maintains and stimulates the interstitial cell of Leydig in the testes to produce the hormone testosterone. As a result, luteinizing hormone called interstitial cell stimulating hormone ICSH. Corticotropes secrete adenocorticotropic hormone to regulate adrenal cortex function. ACTH stimulates the synthesis and release of glucocorticoid from zona fasciculata and zona reticularis of adrenal cortex. Adenohypophyses that contain beta endorphin and gamma lipoproteins that are the peptides and that possibly decrease the perception of pain. And next, pars intermedia, that is a very rudimentary in humans, but in lower vertebrates, especially amphibians and species, it is well developed and produce melanocyte stimulating hormone. And melanocyte stimulating hormone that increases skin pigmentation by causing dispersion of melanin granules. Now, what are the cells, types, and hormones of the neurohypophysis? The posterior pituitary actually is the extension of the hypothalamus. There are no hormone producing cell in the neurohypophysis, no hormone producing cells in neurohypophysis. And it remains connected to the brain by a multitude of unmyelinated actions and supportive tissue, that is pituitarides. Pituitarides are irregular in shape with many branches and resemble to glial cells. Their nuclei are round or oval, and pigment granules are present in the cytoplasm. The neurons of these axons are located in the supraoptic and paraventricular nuclei in the hypothalamus. The unmyelinated axons that extend from the hypothalamus into the neurohypophysis that form hypothalamo-hypophyseal tract and the bulk of the neurohypophysis. These axons also terminate near the fenestrated capillaries in the pars nervosa. Neurons in the hypothalamus synthesize the hormones that are released from the neurohypophysis. These hormones binds to carrier glycoprotein neurophysin and then transmitted from the hypothalamus down the axons by axonal transport to the neurohypophysis. Here the hormones accumulate and stored in the distended terminal ends of the unmyelinated axons, that is known as herring body. Now, the release of oxytocin is stimulated by basically two hormones are produced from the neurohypophysis: antidiuretic hormone and the oxytocin. The oxytocin, that is also known as the pitocin, the release of oxytocin is stimulated by vaginal and cervical distension before birth and nursing of infant after birth. The main target of oxytocin are the smooth muscles of the pregnant uterus. During labor, oxytocin is released to induce strong contractions of smooth muscle in uterus, resulting childbirth. After parturition, the suckling actions of the infant on the nipple stimulates and activates the milk ejection, reflex in the lactating mammary glands. After impulses from the nipple stimulates neurons in the hypothalamus, causing oxytocin release. Oxytocin then stimulates the contraction of myoepithelial cells around the alveoli and ducts in the lactating mammary glands, ejecting milk into the excretory ducts and the nipple. Oxytocin facilitates the transport of sperm in the female genital tract. Oxytocin stimulates the contraction of gallbladder, intestine, and urinary bladder. Next, vasopressin, that is known as antidiuretic hormone or ADH, that is also known as pitressin. Increase water permeability in the distal convoluted tubule and collective tubules of the kidney, creates more concentrated urine after water is reabsorbed and form glomerular filtrate. It is also released during decreased blood pressure and in larger doses may cause smooth muscle contraction in arteries and arterioles. Vasopressin also plays an important role in social behavior. Now, you see here both oxytocin and vasopressin that consist of the 8 amino acid. But if you count here 1, 2, 3, 4, 5, 6, 7, 8, 9. Why 9? I mentioned that they consist of 8 amino acids. You see here 2 sulfur-linked cysteine molecules that are considered a single amino acid, that is cysteine. Now, what is the difference in between the oxytocin and vasopressin? If you see the 3 position in case of the oxytocin, that is isoleucine, and whereas 8 position, that is a leucine. In case of the vasopressin, the 3 position consist of phenylalanine and 8 position that consist of arginine. And vasopressin is again categorized into 2 categories, especially arginine vasopressin, that is found in most mammals, and whereas the lysine vasopressin, that is found in pig and hippopotamus, the 8 position, that is called the arginine vasopressin, whereas that is found in the hippopotamus and the pig, that's the 8 position that consists of the lysine vasopressins. And another vasotocin, that is also octapeptide that increases water absorption. So what are the control of the endocrine secretion? The pituitary gland that consists of the adenohypophysis and neurohypophysis. The posterior pituitary hormones, that is the vasopressin and oxytocin, that are secreted in response to neural activity. Secretion of anterior pituitary hormones that are controlled by releasing hormone and also inhibited by inhibiting hormones from the hypothalamus, which are transported to the anterior pituitary through hypothalamo-hypophyseal portal system. Now, the rate of hormone secretion, that is influenced by a combination of the hormonal modulation, such as feedback mechanism, permissiveness, antagonism, and neural modulation, such as sensory inputs and clock mechanism. Neural activity also governs the pulsative release of the hormones. That is a control of the endocrine secretion. Now, these are the reference books. Tiffard's Atlas of Histology with Functional Correlation, Campbell Biology, Histology, a text and atlas, Baylis textbooks of the histology. Thank you.